What's up, guys? How's everybody doing? Today we're talking about lipids, and we've been talking about lipids, but I want to focus today on the role of lipids. All right, but going a little bit back, let's look at the family of lipids that we have. So, the family of lipids. So, this is a nice figure that I found that shows this. But at the top, you can see that we divide lipids into fatty acids and steroids, right? These are the easy ones, and you know that steroids have a very distinct structure that's very different from those fatty acids that have a polar head and a non-polar tail. And when you have fatty acids, then they combine together with glycerol to give you triglycerides, right? And then they also combine together to give you phosphoglycerides. Whenever you have a phosphate groups, you also have waxes that are esters of um, fatty acids. You have sphingolipids, and these are the ones that have a sphingosine group. And then you have glycolipids and sphingomyelins that are parts of sphingolipids, and the glycolipids we have cerebrosides and gangliosides. This is a nice way to characterize this and classify this, and it's a good figure that puts all of it together. So I like it. But going back to what they do, everybody knows that the lipids are involved in the membranes, right? And we know that the characteristic features of lipids that we've already looked at in terms of what they have in their polar heads or even what they have in their tails, right? Influences how they're embedded in the membrane, right? Uh, their saturation of or unsaturation levels is also going to be a factor. So I gave you some of the examples of fatty acids that are involved, such as these phospholipids and glycolipids and cholesterol. Yeah, cholesterol is a big component of the membrane as well. And as you can see, it kind of invades, the blue one. If you look at the figure that I have, the figure on the right, the blue ones in the membrane are the cholesterol molecules, and that's how they incorporate themselves there. Glycolipids will have a sugar molecule that's outside the membrane. So this is a snapshot of how this is set up, but we're gonna have a whole chapter about this. Don't worry about me not getting into details. But let's talk about lipids as metabolites, right? And as um, as biological signals. I mean, so lipids are thought to just play structural roles in the membrane, but they also act as signal molecules, right? So some characteristics of these molecules, they usually act locally, either within the cell or nearby the cell, and they initiate a cascade of different reactions, just like other metabolites and signaling molecules do. The lifetime of these signaling molecules is very, very short. So um, they are involved in very short reactions. But we have a whole area of the creation and breakdown of lipids, right? And this is carefully regulated, timed. So lipid biosynthesis is a big area right now, and I'll talk a little bit about it in a minute. So actually, let's talk about it. Lipid biosynthesis, right, and breakdown. So this is an example of a lipid pathway. you can see how it's all connected. The synthesis of different lipids, the breakdown of different lipids. So we do need lipids from our diet, but the lipid that we eat is converted and broken down and, you know, used to synthesize the lipids that our body needs. So this is what we call the lipid pathway. Some of the things that you notice on this pathway, I won't have you memorize all of this, but you'll see some of the familiar ones. One thing to notice is the in the middle you see arachidonic acid. You see how it's in the middle. Now you understand how arachidonic acid is important. It's one of the most important acids, but there are other ones as well. But this is the lipid pathway, right? So in the pathway, from the pathway, there are other ways to break down lipids, and one of the most popular one is phospholipases. Phospholipases are, when you hear lipase, it's an enzyme that breaks down lipids, right? Phospholipases break down, well, phospholipids or glycerophospholipids, right? So these enzymes, as you can see in the figure here, they come in and they break them down at either the ester level, right? You can see that some of them, for example, phospholipase A, right? Phospholipase A2 is breaking it at the ester level on this carbon, but phospholipase C and D break down these oxygens from phosphate. So phospholipases are very, very powerful in breaking down lipids, and they can also be dangerous. How dangerous? Well, how about snake venom dangerous? All right, so some of the snake venoms have phospholipases into their the cocktail that they have, right? And those phospholipases break down pretty much, the victims' lipids. But it's not just phospholipases; they have other things as well. All right, so an example of lipid biosynthesis and breakdown, glycerophospholipids can be broken down to produce a variety of different signaling products. So arachidonic acid is one of them, lysophosphatidic acid, diacylglycerol, inositol phosphates. These are all familiar molecules that are involved in the cell, and you can take one glycerophospholipid and break it down, right? So this is an example of how it looks like, where you take the phospholipid, right? The glycerophospholipid, right? And then you break it down. You see how you can break it down into arachidonic acid. Into these ones also gives you prostaglandins and other molecules that are needed in the cell, right? So this is an example of a lipid pathway, an important lipid pathway. All right, so speaking of these lipids, another important molecule that we'll talk about more about how it does this is glycerol, right? I mean, not glycerol, cholesterol. Now, cholesterol, we've seen that cholesterol is not a good molecule. It's not a very good molecule, and it's not a very safe one. It's very crucial, and it's, you need it. We need cholesterol. But when you get too much, that's when you get a lot of heart disease and a lot of other problems. And usually where it comes from is cholesterol just clamping together. Because whenever it clamps together, whenever you have too much of it and it clamps together, it goes into your arteries and then it just blocks the blood circulation, and whenever it accumulates, it can accumulate to the point where it blocks the vessel completely, and then you get a heart attack. All right, so that's why cholesterol is important, but can be dangerous, and it's involved in a lot of people's death as we speak. So because of that, there are ways to kind of counteract the effect of cholesterols, and one of the ways is using a cholesterol mimic or a cholesterol analog, meaning something that looks like cholesterol but it's not cholesterol and it doesn't have the effects of cholesterol, right? So an example is these plant mimics that can be pretty good that you can use instead of using something that's high in cholesterol. And how do they do that? They mimic cholesterol so that they can go and bind to the cholesterol receptor rather than your cholesterol binding, right? So another one is this drug, right? Is drug that they use. So you take this drug and then uh, it binds to the cholesterol receptor instead of the cholesterol, and it reduces the amount of your cholesterol in the body. So this is a graph from your book that talks about how the cholesterol levels go down drastically after you consume this drug. And there are so many other drugs like this um, that are cholesterol mimics. All right, so another example is sphingolipids. Right? So we talked about sphingolipids, and these are the lipids that have a sphingosine group. So you can phosphorylate the sphingosine to give you sphingosine 1-phosphate. And this is an important molecule that's involved in intracellular effects, right? Or even outside the cell, right? It's also involved in binding receptors outside the cell. So sphingosine is also another important molecule, right? And this is how sphingosine goes from sphingosine to sphingosine 1-phosphate. All right, let's talk about another system. And this is called endocannabinoid signaling system. This is the system that's responsible for getting people high. But it's not just that. People think that it stops right there, but this is a system that we have. Everybody has this system, right? So we have a lot of cannabinoids that are innate, that are made by our body, right? So this system right here is kind of a special system because it works just like any other uh, neurotransmission system. If you've learned about synapses and all that stuff, it works like that, but it's a retrograde. We call it a retrograde mechanism because instead of working like your normal synapse where you add neurotransmitters and then they go to the next neuron, it does it the opposite way. So look at this. You see the synapse right here? It works by going backwards, right? But if you can see right here, look at the picture on the right. You have CB1 and CB2, and these are receptors of these cannabinoids, and you have a lot of them. I have a picture of of a lot of them here, and notice here you have one that we call phytocannabinoids and one that we call endocannabinoids. The endocannabinoids are the ones that your body produces and uses, right? And then the THCs, the CBDs, the cannabinols, these are the ones that are made by plants, and they come and they bind to the same receptors, and they have you this, they give you this, extra effects of being high and all that stuff. But know that these receptors and these molecules are systems that are in your body, right? The endocannabinoid signaling system, right? And because of that, by the way, we have a lot of people that are working on drugs and drug discovery that can treat other things. So it's not just about being high. We've seen that targeting this system can treat many, many different diseases. And a lot of people are working on diseases, especially now that this is legal. They are working on research to target these and do a lot of mimics, a lot of modifications of these compounds that I just showed you to find the analogs that might have a really good effect in your body. All right. So another good example is this fingolimod and multiple sclerosis. And this is a sphingosine, right? Fingolimod is a sphingosine 1-phosphate mimic, right? And it has been used, approved by FDA, right? It has approved by FDA for multiple sclerosis, right? So it's a lipid mimic that has been approved to treat MS, and MS is a very, very devastating disease. Some of you have seen somebody with MS. It's not a good disease, and this disease attacks the immune system, and specifically it attacks myelin, right? It attacks myelin. So with the myelin, right, when it gets degraded, that's how your brain gets your brain condition gets it deteriorates, and because of that you start to have cognitive problems, and that's how multiple sclerosis manifests. So phenomyelin, right, what it does is it's going to prevent the lymphocytes from migrating to target tissues. And this is important because the neurodestruction that happens in MS is carried out by these lymphocytes. So whenever you prevent them from migrating to target tissues, well, you reduce the disease. And this has been approved to treat it, right? And this is the fingolimod. Can see how it has the sphingosine the sphingosine group that we already looked at. All right, so if you look at the lipids that you have in the body, right? I think I have made a case that it's really, really important, not just for cell membranes, but it's involved in everything. And a lot of diseases come from the disruption of some of these systems that we just looked at. Some of these lipid biosynthesis, lipid breakdown, or just lipid signaling systems, right? A lot of diseases results from the disruption of these pathways. And because of that, you gotta study the lipids. It's not just about the study of proteins or DNA. You gotta study the lipids in your cell as well. And studying the lipid, right? You can just take a whole cell and study the whole lipids, and not only the lipids, but the amount of lipids in there. And the study of this is called lipidomics, right? Just like genomics, you have lipidomics, and guess what? Just like a genome, we have a lipidome, and one cell can contain about 1,000 different types of lipids. That's how crazy it can get, right? And we have to study what the lipids are, how much lipids are in there, and then you can find the global lipidome of the patient so that you can treat them better. All right. And you can do it, you can even go further and do this in the brain. So, because we've seen that Alzheimer's, for example, but many diseases, when you look at, we call it metabolic analysis, you analyze the metabolites that are changing, right? For example, in an Alzheimer's patient, it's not just the proteins that are changing. I remember we talked about how Alzheimer's comes from accumulation of protein plaques in the brain, but you look at lipids and the lipids are also changing as well, right? So because of that, some people actually use the analysis of the change of these metabolites like lipids as an early warning system for Alzheimer's disease, because sometimes some of these show up before you start to see the changes in the protein. That's how crazy it is. So to do this, you can use mass spectrophotometry, especially like a three-dimensional mass spectrophotometry can give you a 3D map of your brain in terms of where your lipids are and the amount of them. And people do that as we speak, just analyzing the lipids, because usually we're used to analyzing the protein expression, DNA, RNA, but people are using lipids and sugars as well to look at the problems in somebody's brain, in somebody's cells, somebody's kidneys, and so on and so forth. All right, so this is how an MS of the brain is going to look at. So you can look at, the bottom figure will show you the red spots are where you have a lot of accumulations and so on and so forth. And you can use that as, a guide for the patient, right? And if you put it together with the information you have about protein expression, it can give you a better picture of what this patient is suffering from. A few other things that lipids do, you can pause this video and look at this, but you have lipids that are involved in different functions everywhere. I have another table as well, but the point is that they are involved everywhere. One thing that I also want to talk about, we love to talk about this. I actually want to do this in the lab, but saponification, right? Lipids are involved in the saponification equation, and why does that happen is because a lot of dirt is lipids, right? And because of that, you need soap. How do you make soap? You take your lipid, most of the time a triglyceride, right? You take a lipid and then you add a base, and guess what? The base is going to come and remove the acid part. Remember that these three acids are the ones that we added. Let me move this so that you can see all of it. So we remove the acid part. We left with glycerol, and then we left with the fatty acids that we brought in bound to Na, or bound to, it may be, usually the general equation we usually have M or H because the metal may be K or something else. We usually use NaOH, but you can use something else to make soap. So you get glycerol and soap. And as you can see, this soap, right, is still going to be something that has a really long non-polar tail and a hydrophilic head group. See how that looks like? That's the soap that we made. And the soap, as I was saying, the soap is good at isolating dirt, because you have oil and dirt. You have a lot of oil in the dirt. A lot of dirt will be non-polar, right? A lot of dirt will be non-polar, and because of that, our non-polar or hydrophobic tail will come and isolate the dirt. So this is like a dirt particle. It's going to be isolated so that the hydrophilic head is going to be interacting with water, and then this is going to be isolated. And you do this a million times, and that's how your soap works. All right, I think that's enough about soap, because you can say so much about soap, but all you need to know is the saponification equation and how this soap form micelle, right? So this right here, what we do is a micelle. And by the way, you got to remember that we always write down micelle like this as 2D, but this is not a square, is it? It's a sphere, right? So the lipids are not the lipids, but the soap molecule are all around the particle, so they surround it. And this is a better representation of it here. As you can see, the one on the right is a simple micelle, right? And it's a sphere. But we know that the lipids not just form micelles, they can form even much more intricate uh, structures, such as the one on top is how the cell is made, right? They not only form a micelle, but they form a bilayer, right? And the bottom part shows you like a linear portion of the bilayer, but the bilayer is formed by, well, the nonpolar groups are facing the inside, the polar heads are facing the outside. That's how you have a water environment, a polar environment on the outside of the cell and the inside of the cell, and we're still good. And by the way, this example right here is something that we can do in a test tube in the lab, right? It's if you put these lipids in the same conditions, they are going to give you the same or similar shape, right? So you can do this in your lab, no problem. All right, so actually, an example of doing this is the COVID vaccine, right? The COVID vaccine is made by, at least the mRNA vaccine is packaged in a lipid container like this one, where you just put the mRNA inside, you make a lipid, and then you make a lipid container, and then you deliver it. And why is it important? Is because if you just add mRNA, it's going to be destroyed, right? If you just inject somebody with mRNA, it's going to be destroyed right away. So you envelop it into a lipid, and then you just deliver it. It's an amazing way to deliver drugs, and it's not just the mRNA. It has been done many times. Let me see if I can find a nice figure of how they do it in the vaccine. There you go. So this is how the mRNA vaccine looks like. This is the thing that they injected in your arm. It has the mRNA inside, but on the outside, it has a phospholipid layer, right? And it can be a bilayer or a monolayer. But I thought it was really amazing that lipids are the ones that actually allowed the vaccine technology to pretty much be developed in less than a year. But that's enough. That's enough about lipids. We'll talk about the membrane next time. Bye.